I would like to talk about how climate change actually hits the ground or hits the water in the context of all the other things that are going on. And I'd like to relate what I've learned in the last few years through three stories. A story about coral reefs, a story about tropical Great Lakes, a story about Stellwagen Bank right off the coast here. The coral reef work has basically taught us some things we wish we hadn't known about the frequency and intensity of climate anomalies and how they affect aquatic systems. In the Great Lakes, we found out that downscaling doesn't always work, that very, very strange climate effects can emerge that you couldn't possibly have pulled out of the global models. It requires an enormous amount of idiosyncratic information. And offshore here, climate change may have been invoked erroneously, where the real effects are still occult. So we'll take a look at these quickly, and if I run out of time before I run out of stories, I may cut ahead. All right, coral reefs. This is coral reef in Fiji, and I try to spend as much time as possible doing this instead of being here, and my chairman has noticed. Coral reefs are in a very bad way. All over the world, they've been degrading since about 1980, very, very rapidly. Degrading means the coral dies, falls apart, disintegrates, the fish go away, and most of the services, most of the good things people get from the reef ebb and eventually disappear. This process has been called by my old advisor, Jeremy Jackson, a slippery slope to slime. But what Jeremy did not mention at the time is that the skids that these reefs are sailing down are greased by climate change. It's the interaction of climate change and local human impacts that create the picture that we see. The situation is pretty horrible globally. This is a stoplight map from Reefs at Risk, a report that we do every 10 years or so. And all the reds are reefs that are not any longer recognizable as such. In other words, there's still a little bit of relief there, but if you swim over them, you don't see a lot of coral and you don't see a lot of fish. The reefs that are in the best condition are in the coral triangle. And those reefs show an extraordinary resilience despite very high human population density. And we're just trying to understand.
Green or yellow?
Green and yellow. Like, what? Green is really good.
Okay, green is best.
Yeah, yellow is watch out, but still okay. Red is, it's not even a coral reef anymore. So we'd like to understand why those reefs are still doing fairly well despite being dynamited very frequently. And we'll talk a little bit about that in a minute. But this is the trajectory for percent live coral cover in the tropical North Atlantic, so the Caribbean basically, and Florida, between the 70s and 2002. And I can report that there's a very slight, barely significant uptick as we go toward 2015, and we began to realize what's going on. Now, when the coral goes, the fish stay around for a surprisingly long time, but there are a few species that are obligately tied to particular corals, and they disappear right away. This is a tiger grouper, which was one of the iconic fish of shallow water reef crests all through the tropical Atlantic. They're pretty rare now, and their home, the elkhorn coral, Acropora palmata, was wiped out along with staghorn coral, and they've diminished in abundance throughout their range. The whole tropical Northwest Atlantic, they're so diminished that they're listed as threatened on the U.S. Endangered Species List and endangered on the IUCN World Red List. This is tantamount to cement being declared endangered. This is the fundamental building material of all the North Atlantic reefs. And these things are down to scraps now. Tiger grouper disappeared. Schoolmaster snapper found an alternate habitat. The prop roots are red mangroves. Now, elkhorn and staghorn coral deserve special attention. They're two corals, and they're hybrid, which behaves as if it were a species of its own. that grow very, very quickly and are basically the predominant builders of everything above 8 meters throughout the entire tropical West Atlantic. So they build the part of the reef that protects tourism. They are what break the waves. But beginning around 1980, we started to lose them wholesale. This is a field of pre-1980 coral on Carysfort Reef in Florida, and this is after a major thermal anomaly. The algae that are symbiotic with the coral are expelled or they leave, and the coral, if it has to remain this way for more than 5 days, starves. This happens at a temperature of about 30 degrees centigrade. The optimum temperature for coral growth in this part of the world is 29.2. They have no headroom. All it has to do is warm up above 30 for 5 days running, and the coral is dead. We've seen this happen in deeper water too. This is a before and after picture of elkhorn coral, exactly the same spot in Florida. There's a guy, a former Globe guy named Dave Arnold, who has this big project called Double Exposure. He's going all over the world and photographing places that were photographed very well in the mid-70s to see how they changed. This is part of my study area on the Fore Reef in Jamaica in the late 70s when I was doing my dissertation work. This is the same spot now. It's all seaweed and sponges. And this has happened on a grand scale throughout the range of the coral. Now the problem is that the local stress through most of the northwest tropical Atlantic is so great, from overfishing, from deforestation, from pollution, from mining of corals to make concrete, that it almost doesn't matter what the climate is doing. But climate change for these corals is the coup de grace. Now, in order to find out how corals will deal with climate change in a place where there are no local impacts, we had to go somewhere there were no people. So the New England Aquarium led an expedition in 2009 to return to a place they'd visited 7 years earlier called the Phoenix Islands Protected Area. It's dead center in the middle of the Pacific Ocean. It's the most remote place on the planet. It's a 5-day beat through hellacious seas, 20 foot the whole way. But when you get there, it's worth it. The place is just spectacular. So this is a picture from 2002 that was taken on one of the atolls that we've been working on. And this occurred 3 months after that picture was taken. This is obviously a different coral field, but it's entirely bleached and dead. That happened because the most severe thermal anomaly yet recorded sat over the Phoenix Islands for 7-degree heating months. That means for a total of 7 times about 30 days, the temperature was lethal for the coral-algal symbiosis. The place was toasted. The question was, given that there were only 24 people living on this island and nobody on most of the other 7 atolls in the system, could the coral recover if there were no local impact? It's a test of the synergy hypothesis. Well, when we came back 7 years later, the place was absolutely covered with this pink stuff. This is coralline algae. It's a seaweed, but a special one. It secretes a hard skeleton, much like the coral. It's made out of a different material, high magnesium calcite, which, by the way, is quite soluble, instead of aragonite, which is a more resistant form of limestone. But this material acquires a layering of bacteria that secrete chemicals that are come-hither messengers for baby corals. Coral and algae is an indicator that the environment is perfect for corals to grow and survive, and here are all the baby corals coming in. Major players in this story are parrotfish and other herbivorous fishes, because if they were not there in enormous abundance, you wouldn't see any of that pink stuff. It would all be fleshy algae, seaweed, or a kelp, covering that space. It's also thought that large sharks and big animals like sea turtles and dugongs also play a role in the resilience of coral reefs but that's controversial. You'll hear it repeated over and over again that you need big sharks to have a healthy reef. Well, you need big sharks to have healthy reef tourism. But the relationship between top carnivores and reef health is still obscure. The thought, though, is that these large predators, by preying on smaller fish, favor rapid growth to large body size in parrotfishes and surgeonfishes, making them better lawn mowers. They keep the fleshy algae down, the coralline algae comes in, and in 7 years, it's as if you press the reset button on the system, which is really great. The problem is, how often are we going to see those devastating bleaching events? If they're more often than once every 7 years, then even the most pristine coral reef on Earth will not survive. It'll never quite make it back. In the Caribbean, it's a different story. After the big die-off in the 1980s, here's an elkhorn reef crest from Belize, before and after. People began to realize that the reefs were not recovering. After 7 years, after 20 years, after 30 years, they were not coming back. They're just now beginning to squeak a little bit toward recovery. And the reason is apparently that the cumulative local human impactors are at such high levels that, combined with the periodic warming and the flushes of disease, usually from human sewerage, that are accelerated by the warm water, there's not a chance for these reefs to come back until you start reducing those local impacts. And in Belize, we've tried. We've had a campaign going for several years to try to end the fishery for herbivorous reef fishes. A lot of these were speared on snorkel. And so we had a campaign called Real Men Use Their Hands. And I don't know if you can see it back there, but there's a guy holding a little hand line, and his paramour has his spear gun at a safe distance. And of course, use their hands for what, you know? But the church did not like that. But it was very effective. The other campaign we had to mount when 2 Jamaican investors came to Belize and pushed Jamaican fish traps, which are extremely dangerously effective at catching parrotfish. They tried to push them on Belizean fishermen, saying that they promised to buy all their catch, and they gave them the traps for free. So we had to mount a national campaign in Belize to stop the traps, and now there are no such traps allowed in Belize. And in fact, you're not allowed to catch a parrotfish there anymore, and it's helping. It's already apparent that it's working. In addition, there's an intrepid woman in Placencia, Belize, who's been growing and putting coral back out on the reef. An enterprise that might seem Sisyphean, but in this new environment of better stewardship, it's working. By the way, there's a parallel effort in Florida. I have a student working on it. She just showed me the demographic data on all the corals that have been put out on the Florida reefs, and it's dismal. Something is still wrong enough there that cultivating and gluing corals to the reef is simply not working. And the question is, what spells success? What is it that a reef needs to work? And we have a broad array of reefs to compare to address this question. In the Indo-Pacific, there are almost 280 species in a single family of hard corals, the Acroporidae, the staghorn and elkhorn corals. And those reefs are fantastically resilient. In the Caribbean, there are just 2 species, elkhorn and staghorn, and a hybrid swarm that unites them genetically. Is that the reason the Indo-Pacific is more robust? Is it just that diversity can actually matter to community resilience? Well, we hope to find out, but what's clear is that among the differences between the resilient and non-resilient reefs is species diversity. Never before in the entire history of reefs on Earth, which is a history that goes back almost as long as living organisms, never before has this been the case. We've had high diversity reefs and low diversity reefs, and in the geological record, they last just as long. So something new is going on. The other idea we had is that we might be missing the most important diversity, that even though species diversity may be low in the Caribbean, there was a lot of genetic and adaptive diversity within species. In fact, we may not even know what a coral species is in this ocean. These are all staghorn coral, but they look wildly different from each other for one of two reasons. Either they have genes from elkhorn coral that's gotten into them, or there's just a lot of genetic diversity in staghorn coral in the Caribbean. It's as if this one species is behaving like 20 or 30 distinct forms. So now that we have the molecular tools to look at this dark diversity, we're doing it. We're having farmers put out all the different genetically distinct forms and look at their relative success. And it's not just the coral. The algae that are symbiotic are phenomenally diverse. The microbiome, there are about 20 or 30 species of prokaryotic cells that are habitually associated with any particular species of coral. Coral, the word, is meaningless without the symbiotic algae and the symbiotic bacteria. Now that we can look at the entire organism, this little community which we call the holobiont, we're beginning to understand corals a little bit better. And we're beginning to understand why things go wrong. This is a staghorn coral attacked by my favorite love-hate partner, this little fish, a damselfish, that kills the coral and grows seaweed in its midst. It's a highly territorial damselfish, and the coral, any staghorn coral anywhere in the Caribbean, has to put up with these bastards. That means that the life of a staghorn coral is one of constantly healing nicks and scrapes. Add acidification. How well will they heal those nicks and scrapes? This is a coral that has an immune response to this little hermit crab that's taken up residence there. There are hermit crabs that stick their butts in holes in coral. It's amazing. Anyway, the immune response is visible because a chemical is produced as part of the immune cascade that is brilliantly fluorescent. So in this coral, we can actually study the immune response without sequencing its DNA or looking at its messenger RNA. We can see gene expression in fluorescent day-glow pink. This is a coral reproducing, which is the time of greatest stress for these corals, even when the water is cool. And these are the little zits that the damselfish elicits in the host staghorn coral. And they form because when the fish bites the coral, there's a little area of no coral tissue for a couple of weeks. Algae gets in, and then the coral tries to grow up around it and seal it off, and it creates these structures called chimneys. Anyway, I lead a group called the Coral Whisperers. There are 6 labs that are doing gene expression profiles on corals subjected to different kinds of stress, and we're hoping to use those methods as reporters for whether attempts to reduce local impacts have succeeded. The best one to ask is the coral themselves. Are they happier? Look at their messenger RNA. All right. In Brazil, there are also coral reefs, but they look very different. And the entire economy of Bahia, Brazil, is about the beach. Half the population of Brazil goes to the beach every summer, no matter where they are, even from the Amazon if they can afford it. And on the beaches is everything you want: food, drink, other things, hotels, and the hotels are called pousadas. This is the Itacolomus Reef Complex off of the town of Caravelas, that's Ponta de Baleia. This is an area where astoundingly there are coral reefs and humpback whales. And it's a fantastic tourism draw. So this is why people are going there. The problem is the beach is going away and the reef is going away. The reason this is happening, let's think about the beach first. This is a map of a tiny part of Brazil, that dark spot up there, right where we're working. And that is a map of all the reservoirs in this small area of Minas Gerais. Those are dams in the rivers, all through the river system, like this one. And what they do is retain sediment. and prevent it from reaching the shore. So what they're doing is starving the beaches. So the beaches are threatened initially because it's hard for them to get sand. Then on top of everything, in the unusually warm summers that Brazil has been having, there have been outbreaks of a strange disease that we call white plague that turns out to be the result of a consortium of about a dozen different microbes that together attack and kill the coral. And this isn't just any coral. These golf balls are Musasmilia, a genus of coral that's been extinct everywhere on Earth except Brazil for 3 million years. The entire coral reef system of Brazil is a living fossil. These corals exist only in our study area, a region of perhaps 10,000 square kilometers, which is not very big, and these are the major reef builders. As the reef dies and begins to collapse, the water can surge over the reef crest and further erode the beach. The critical window, based on the geomorphological studies we've done, is 10 centimeters clearance. If there is consistently 10 centimeters clearance at mean low water, the beach will go away. The reefs are holding the beaches in place. What's holding the reefs in place? Well, there's a large parrotfish that makes up to half or 2/3 of the biomass of the fishes on these reefs. It's called Bougeon azul, Scarus trispinosus. It gets about the size of this table. Most parrotfish taste like shit. This one is delicious. And it supports a cottage industry of spear fishermen who shoot the parrotfish to feed the tourists who are there to enjoy the beach, which only exists because of the coral reef, which only exists because of the parrotfish. They're shooting themselves in the foot. So we began a lengthy study of this animal and looked at its distribution and tried to determine if protected areas that were put in place to build up the parrotfish population were working. It's hard to see this from the back, but these tall bars are not protected reefs. They're just reefs that are so friggin' far away, nobody can afford gas to go out there. That's the only thing that has been effective in protecting these fishes. We're about to put a bill into the Brazilian parliament to make it illegal to kill these, but we didn't want to hurt the artisanal fishermen who have no other source of livelihood, because these could be fished sustainably. But it's like a house of cards. If you don't manage the keystone properly, then the vulnerability of the entire system to advancing sea level rise and warming seas is elevated exponentially. In order to get the whole story, we backed up and created a project funded by the German Climate Initiative called ICI, which has been going on for several years. And we're looking at the entire landscape of eastern Brazil in the province of Bahia, including the Atlantic rainforest, the eucalyptus plantations, these weird marshes that are made out of bromeliads, the coral reef, and several other environments. And beginning in 2011, we began a massive exploration of the part of the continental shelf adjacent to the tourist industry beaches. And we discovered things we could not have dreamt of. This is an out-pimple of the shelf, the continental shelf in Brazil, that extends offshore about 100 miles. And it's about 150 miles north to south. Here's the reef with those golf ball corals. But we also discovered vast areas, thousands of square kilometers covered by kelp forest in the tropics. How weird is that? Hovering above those forests were massive schools of a reef fish that serves as an important energetic link between the reef and big predators that people like to eat, like groupers. But we discovered some things that are even stranger. We did a sonar survey of this entire upper portion of the Arroios Bank, an initial exploration in what was a paleo lake at a lower sea level stand. We now have cores from in there. And we discovered that about half of this area is covered, you're not going to believe this, by algae balls. Algae balls made out of coralline algae, that hard algae, remember? It is such an enormous area that this is actually a significant carbon sink. And we began to think, maybe Brazil can get carbon credit for not destroying this area. And that thought occurred to us just about the time that the sugar industry decided to mine these things in order to get lime to sweeten rainforest soils so they could extend the sugar farms. So we're still fighting that battle. This is a heat map of the rate of carbonate sequestration as you go across this area that's covered by these rhodoliths, or hurlito. They're algae balls about this big. I should have brought one in. Each one is 300 to 400 years old, and they've been piling up for the entire Holocene. There's an 8,000 to 10,000 year accumulation of these things, which amazingly pile up about the same speed that a coral reef grows. And then we found something even stranger. In the middle of our sonar survey, we found these deep holes. And we asked the fishermen what they are. They're holes about 20 to 30 meters deep from a platform that's already 40 meters deep. So they're a little hard to dive on. So I dive and point for 5 minutes and then go up and decompress. But our grad students are sent with mixed gas, so they can stay down there. As long as they don't die, this is a great system. And what they did is go into these holes, or buraca. Buraca actually means something else too, but I'll not go into that. Anyway, these buracas are geological mysteries. How did they form? They don't resemble sinkholes. It turns out that the most likely mechanism of formation is that any little depression in the platform fills with seaweed, which rots and acidifies the limestone and burrows down into it. And this is us hovering over a buraca at a depth of about 130 feet, and it looks like a sea of kelp, but you can actually go straight down into it. And underneath is an anoxic column of water that is remineralizing the kelp and providing raw bioavailable nutrients to the next generation of kelp and coral. So look at what we discovered just by looking around. A horrible problem with a cascading crisis going on the beach, and a huge carbon store we didn't even know existed offshore. All right, story about the Great Lakes. This is Lake Malawi, and these are the famous cichlid fishes. Similar fishes exist in Lake Tanganyika and Lake Victoria. But they're going extinct in Lake Victoria very rapidly. But that's typical of freshwater fishes all over the world. This is a graph of just the number of fish species, and they go from very vulnerable to extinction all the way to they'll probably benefit from the things we're doing to freshwaters in California. And as you can see, all the native ones are at death's door. This is a graph of the cumulative number of extinct taxa in North America over decades from 1900 to 2010. It's finally leveling off, just barely. You think that's impressive? In Lake Victoria, we think there were 600 species of endemic cichlid fishes that occurred nowhere else in the world, each adapted to a particular, very specialized mode of life. 70% of them are now missing in action. And the reason for this has become part of the popular media and a movie that almost won an Oscar called Darwin's Nightmare. Has anyone seen it? Yeah, you saw it. You were subjected to it. Well, what do you remember from it?
It was a nightmare. It was a very complex system involving many people and countries and violence and a cascading effect of not so good stuff.
Yeah, when Hubert Sauper, thanks Em, when Hubert Sauper went to Africa to make this movie, he found me on the web and we became friends, and he was determined to go to Lake Victoria to tell the story of the cichlids, of their fantastic evolution and Extinction. But you look at the movie is about the arms trade, prostitutes, and AIDS. Because what he discovered was that the introduced fish, which you see here being smoked en masse, it's called the Nile perch. It reaches a length of 2 meters and a weight of about 100 kilograms. That fish is perfect to produce white fish fillets for Europe. It was introduced by the British, and it was thought that Nile perch was the reason for the mass extinction of cichlid fishes. And we formed a team and set out to try to puzzle out what was going on. And at first, we tested a hypothesis that Nile perch had indeed eaten all the little fish, and that was the problem. And based on that, we set up a fishery model that said, if you fish Nile perch, this red line, at a certain rate, you can see the haplochromines come up, and there's the sweet spot. You have to keep fishing at that level of intensity, and then the two could coexist. And we tried to institute this in the fishery laws of Uganda, Kenya, and Tanzania. Now, we arrived during a particular climate regime on the lake where there were very low winds and it was extremely warm, and we naively assumed that's always the way it was. Under those conditions, the lake had begun to stratify, to layer, and there was no oxygen below a depth of about 25 meters. The entire 3/4 of the volume of the lake could no longer support fish. We had to explain that, and we explained it with another top-down hypothesis, that the perch had eaten all the fish that eat the algae went to the bottom of the lake and rotted, the warming surface waters stabilized the layering of the lake, and that's how we got to that situation. Except we were wrong. More recently, based on evidence that we had accumulated over several decades, we invited another oceanographer into the mix. Her name is Sally McIntyre. Her problem was to explain why we went from diatoms to toxic blue-green algae, from cichlids to Nile perch, and from giant zooplankton to tiny zooplankton. And what she found out is when she projected wind speeds over time across the surface of the lake in that upper graph, there is a period represented by this gray bar several years long when the winds died. Those are the conditions under which the mixing of the lake stops and it stratifies. We had it completely backwards. The winds died, the lake stratified, a huge proportion of the cichlids were suffocated, and then they could no longer do a thing that we hadn't thought about. Baby cichlids eat larval Nile perch. Adult Nile perch eat adult cichlids. They were chasing each other's tails. The haplochromines are what kept the Nile perch in check, incredibly. When so many of the haplochromines died, the Nile perch erupted and became this enormous export fishery that they are today. So in this particular instance, a local climate effect that can be traced to general changes across the continent of Africa, but has this unique local impact, is what caused Lake Victoria to undergo a phase shift, a flip. We never understood that until just a few months ago. I want to talk a little bit about New England and coastal banks. This is what Fijians call a vanua. This is a watershed system with people living in it, a coupled human and natural or social ecological system. Traditional societies have understood how these things work for a very long time. We just sort of ignore it. In New England, the food base for all the large animals is a group of small silver fishes and squid and invertebrates called forage species, or colloquially forage fishes, and herring. Sea herring is a big one, and also river herring. Herrings that look the same as ocean herring, but instead of spawning at sea on the sea bottom, they run up all the rivers of New England. The herring runs in New England have been destroyed. How? Well, they can jump pretty well, but they cannot jump 10 meters. the dams on all these rivers that date back to the original mills that were built on almost every stream in New England, abbreviated, just arrested the anadromous fish runs and reduced by at least 95% the availability of forage in the nearshore waters. So whales and cod alike were starved by the damming of the rivers. And it's something we forget because it happened a while ago. Meanwhile, with the herring that are left, there's a sort of tug-of-war between attempts to protect endangered large animals like the humpback whale and attempts to catch and sell herring, as well as their predators, the cod. In order to elucidate this trade-off and understand how dams and climate change have influenced it, I lead a modeling team called MIMES that tries to work these things out, tries to understand the dynamics of coupled human and natural systems. And in this particular case, there was a special consideration. The Gulf of Maine is the body of water on Earth that is warming the fastest. It's an accident of our local oceanography. So in addition to the dams, we have this runaway warming going on. Now, we were able, to cut to the chase, we were able to construct a workable model of the interaction between forage fish and all of their predators and the industries dependent upon them. So for example, this is a computer game, a graphic user interface that Professor Suchi Gopal developed called Midas. And in this particular case, what it's telling us is that at a particular level of fishing on forage fish, this is a slider, in the month of July, these are the pixels, the areas in which you've lost whale watch value because you're allowing people to catch whale food. Now, these trade-off points move as the overall productivity of the system changes. And as the Gulf of Maine warms, the productivity is going down because those cold water algae and cold water fishes that are geared to an immense burst of biomass in the early spring to midsummer are no longer able to prosper here. Now, as a result of this, Andy Pershing of the Gulf of Maine Research Institute proposed that the fact that cod had collapsed and not recovered, which I'm sure you've all read about in the news, was because of the warming waters of the Gulf of Maine. Climate change was fingered as the culprit. But think about it. What about the 600 years of overfishing and damming that has been modifying the system? We've been working with historical ecologists to reconstruct the appearance of the fishery in Massachusetts over the last 200 years. And what we've discovered, plain as day in the town records of every town hall around New England, is that there used to be an enormous amount of fish just a few steps off the beach. They're all gone. Everything has been pushed offshore. Fishing pressure is probably the primary reason why both the cod and its preferred prey are at such chronically low levels. So here's an example where climate change has been mistakenly accused of causing this collapse. Now, MIMES is an invention of a Dutch survivalist farmer named Roel Boman and an economist, Bob Costanza. Bob has a habit of always leaving wherever he is. He's like an itinerant economist. And as a result, I wound up inheriting this team. And we've been analyzing the human natural dynamics in East Africa, in Cambodia, in Massachusetts, in Brazil, and we're looking at the Pacific now. And we were just quietly doing this. We're pretty obscure. I'm a science fiction buff. So about a month ago, I was reading this new book by my favorite hard sci-fi author, Kim Stanley Robinson. And it's about the first attempt to colonize Tau Ceti, a planetary system 12 light years away that drew the attention of Isaac Asimov, a BU professor, many years ago. As sort of a way of honoring Asimov, Stan wrote this book about a giant spaceship packed with all the biomes of Earth and people living in there for multiple generations so that at a 10th light speed they could reach Tau Ceti. On the way, the heroine, the ship's engineer, is dying of incurable lymphoma. And she's trying to school and groom her daughter to take over de facto leadership of the ship community. But her daughter doesn't like math. Okay, now I'm sitting there reading this book, and the next sentence I read is, the mother telling her daughter, you must use mimes and Midas to understand how the ship works. I'm going like, what? So I contacted the author. He has no idea where he heard about us. I mean, there's nothing else called this. But it was very gratifying to know that our work would one day actually be used, because it had been rejected by the local ocean planning board because they don't like the word trade-off. They don't like the idea that somebody wins and somebody loses. So we will be relevant, but it will take 500 years. Okay, now one more very quick story. We became very interested about 6 years ago in the chaotic dynamics of fish populations and marine communities. You all know about chaos? I mean, most of our lives are deeply infused with it. Chaos is a mathematical property that most natural systems exhibit. They exhibit recurrent behavior, but they never do exactly the same thing again. And the question is, can you capture that behavioral modality without getting hung up on the fact that you can't make perfectly accurate predictions? Well, a colleague of mine, George Sugihara at Scripps Institute, who is a mathematician, figured out how to solve that problem. And he applied it to the world economy, working for Deutsche Bank for, I think, 11 years. And then mysteriously, just before the whole economy collapsed, George left. Just before the collapse, with his money, and went back to Scripps and decided to work on fish again. And together, with a team of about 8 postdocs and maybe about a dozen grad students, we began doing what's called constructing state space representations of the dynamics of the Gulf of Maine fishery and the California coast, because those are 2 places where we had a good 40 years of data. This is an example of a seemingly meaningless time series of fish population size, and that's what it looks like when you plot it up using this method based on a theorem called Tocquen's theorem. What you're doing is plotting lags. What was the system doing now, a year ago, 2 years ago, and so forth? And that captures the velocity and direction of the system as a whole. Looking at it this way, every single species is a window on the entire system, and it allows us to calculate how chaotic are the dynamics, how well can you predict the future using this model, and how much of the dynamics are actually chaotic. This, for example, is the manifold, the spatial representation of an obscure fish from here that we know almost nothing about, but the math reveals that it exists in 2 alternate states. A perfect Lorenz butterfly. You almost never see this. We still don't know what that means. This is an empirical model. But applying the model to hundreds of different time series runs of data, we made a horrifying discovery. If you look at data on the abundance of fishes that are not a commercial target, they're caught only by accident, so they're not being sucked deliberately out of the ocean, and you look at the extent to which their behavior is linear, non-chaotic, or chaotic, you see that those fish that we don't touch mostly exhibit a highly simple and predictable behavior. If we look at the fishes that we're targeting, their dynamics are much more chaotic. If you look at the abundance of the fish at the landing, in other words, they've gone through a sail, there's even greater chaos in the numbers. Now, if you push the chaos in the system far enough, it quickly moves out of that narrow window in which you can still make predictions. It becomes, for all intents and purposes, random noise. You can't predict anything. You can't manage anything. You don't know what's going to happen from year to year. So we tried another test. Emily Klein, one of our grad students, looked at a period of historical data in the 19th century, all the catch data for cod, haddock, pollock, and herring, and then looked at the catch data from now. The dark bars are the level of predictability, in other words, the number of time series that delivered a significant predictive result in historic times, 150 years ago, and today. What this suggests is that our impact on the system is not only degrading it, it is making it unmanageable. We are driving the world into a type of non-deterministic chaos that verges on random behavior. Do you realize the implications of that? It means we are headed toward a complete loss of control. Now, at the heart of this is this dynamic interaction between the things we do on a local scale and our modification of the ocean-atmosphere system at a global scale. It's horrifying, but we know what we must do. Thanks.